seen what our students do and gotten to take part in bits and pieces of their projects and their research and work. It is an incredible achievement on the behalf of these students. And I'd like to start off by giving a real round of applause for that being the case, and one of the things I love so much about IoES and this program in particular is that it is a collaborative and supportive enterprise, and none of these projects would have been possible without the incredible support of everybody at IoES. Cully Nordby, the associate director, Marilyn Raphael, our director, Scott Groover in communications, who gets me out of any number of jams with technical issues throughout the year. Royce Diekman, your student affairs officer, who none of you would have lived through this 4 years without. And then the myriad and incredibly accomplished faculty and lecturers and others involved in supporting your projects, and our clients who bring these to us and provide these incredible opportunities for you. Thank you to all of you for everything you've brought. We really do appreciate it. With that, I'm going to turn it over to our director to welcome everybody today and to sort of get this event started. Marilyn, thank you so much for being here. And everybody, please give a nice welcome to Marilyn.
Good afternoon, everybody, and a really hearty welcome. It's nice to see a full audience of smiling faces on a Saturday afternoon when you could be outside. Welcome to the Institute of the Environment and Sustainability's Senior Practicum presentation. 135 students, 16 projects. I don't know how Noah and those guys pulled it off, but you're here and we're ready to listen. I'm really happy to be here to see the culmination of your year-long efforts. I say year-long, but really, you've been working hard for at least the last 4 years to arrive at this point. And you know perhaps better than I do how challenging a period that was, beginning with COVID and ending with now. Right up to today, the last few weeks, I just want to acknowledge have been challenging for everybody, but we're here now. And you're here because you believed that what you were doing is important and you persevered. This is your chance to communicate to us what you have learned. As always, when I come prepared to this event, I look at the projects that will be presented today. And I'm envious that you have had this experience. I mean, I'm really happy for you. The range and complexity of the projects are truly impressive. I saw projects that deal with conservation in Cameroon and at UCLA, with water quality in Hawaii and in Michigan. Those are just a few, but I also want to say I'm personally interested in the project examining flood susceptibility in Tobago because that's one of the islands where I come from. So I'll be watching. And Sky Chef, I won't think about airline food in the same way again. That's it. I note appropriately that there are continuing strong themes of environment and sustainability and environmental justice. And I want you to know, I'm sure you have a sense, but I want you to know how truly rare it is for undergraduates to be engaging in work that is this impactful while you're still students. You're really fortunate to be able to do this. And the IoES is really happy to give you that opportunity. It prepares you well to take on the environmental challenges we're facing. Know that your work reflects positively on UCLA's role in the community and for pushing science to effect real-world change. Know also that the IoES is very proud of the work that you've done, and I now look forward to hearing you talk about it.
Thank you, Marilyn. With that, I think we should take things away from the staff and faculty at IoES and turn over to the students who are the real stars of this show. And I'd like to welcome our first team who worked with the Aquarium of the Pacific to come up today and start us off. Please come on up, team.
Hello, everyone. First, we'd like to welcome you all for coming today. We're so excited to see all the hard work from all the Practicum groups. Today, we'll be starting off with our group. We worked with the Aquarium of the Pacific in Long Beach on a blue carbon project. The goals of this project were to set up the aquarium to purchase carbon credits that are marine-based in theme with the aquarium, to identify a project site to restore eelgrass meadows, and to develop credits for biodiversity to help improve the ecosystem near Long Beach. Our proposed project timeline starts with an investment in blue carbon to take immediate action to offset emissions from the aquarium. Then we advise identifying and restoring eelgrass beds with the help of local volunteers. Finally, we hope the aquarium will stack blue carbon credits from the restoration site with biodiversity credits, which will confirm the aquarium has improved local biodiversity.
Hi, I'm Lily, and I'll be speaking about the voluntary carbon market. Okay, so what is blue carbon? Blue carbon is a process by which carbon dioxide is absorbed from the atmosphere and stored in the ocean. And there's a couple different types of blue carbon capture methods. We'll be focusing on ecosystem restoration. So when you plant a mangrove tree or restore a seagrass bed, those plants grow and absorb carbon from the atmosphere and hopefully store it for a long time. And the Aquarium of the Pacific is interested in blue carbon because they want to purchase carbon credits to offset some emissions and hopefully invest in conservation as well. And obviously blue carbon has a lot to do with the aquarium because these are marine-based credits and we're speaking about an aquarium. Today, blue carbon credits are not available on the global market, but they will be in about 2 to 5 years. For terrestrial-based carbon, 1 carbon credit is equal to about 1 ton of carbon dioxide sequestered or avoided, and 1 ton is about the size of a fully grown horse. We spoke to the Seattle Aquarium about their carbon credits because they've been purchasing them since 2020, and they decided to purchase credits with the Rimba Raya project in Indonesia, which is a forest-based credit. So they are paying people to replant a forest in Indonesia, and as those trees grow, they're taking in carbon dioxide and hopefully storing it for a long time.
Hi, I'm Mitch. So I'm going to be talking about restoration and the potential for carbon capture. We looked into seagrass restoration and that landscape in California, as well as the variables of seagrass restoration and how you can translate that into carbon credits. So for benefits of seagrass restoration, eelgrass, which is the most popular form of seagrass, is very vital to maintaining a healthy ecosystem. That could be through protecting the coastline and storm surges, as well as nurturing fish and keeping the water clean. And then another factor is it can sequester carbon at a very high rate, 0.83 to 1.78 metric tons per hectare per year. And as Lily just said, 1 ton of CO2 sequestered is 1 carbon credit. So there's a very high potential for carbon credits in seagrass restoration. And then seagrass meadows can be found all along the California coastline, making them a great local option for restoration projects. And that can be seen on the next slide, where we looked at ideal restoration sites that the aquarium can work with and groups that they could work with to potentially both help local ecosystems as well as offset their own emissions and even go as far as sell those carbon credits. These 3 groups right here, the Bay Foundation, the Coastal Resources Management, and the Santa Barbara Channel Keeper, have all done seagrass restoration in the past, but none of them have done seagrass restoration that turned into carbon credits. So that is something that the aquarium would like to do in the future.
I'm going to be discussing biodiversity credits today. So biodiversity credits are an economic instrument that allows private companies to finance activities that deliver net positive biodiversity gains. This is important because biodiversity credits can increase funding to important things like conservation and restoration of vital ecosystems. Companies that invest in these biodiversity credits can gain recognition for improving the ecosystems and be recognized for their environmental contributions. How are we going to do this? I will be discussing stacking and what that means later in the presentation. But what's going on in the world? So globally, biodiversity credits are not very systematic, and a lot of different companies use different metrics to measure them. So one company might look at hectares, and that'll be what they use per biodiversity credit. Another might look at apex predators and see, once an apex predator returns to the site, is this a good indicator for improved biodiversity? There's also mitigated and voluntary biodiversity credits. Mitigative would be if a company damages some ecosystem and hurts the biodiversity, they then need to offset that impact by improving biodiversity at a different location. What we're focusing on is voluntary biodiversity credits, where companies voluntarily are aiding the ecosystems. There's also terrestrial and marine biodiversity credits. Terrestrial tends to be easier just because it's much simpler to restore and quantify biodiversity on land. Whereas in marine environments, it's an emerging market and we don't exactly know what to do yet, but that's what we've been working on. So we've been looking at Vera and Plan Vivo as two companies that we could potentially collaborate with on the biodiversity credits. Vera is known for their certified nature positive investments in their biodiversity credits, and Plan Vivo has their own methodology that ensures that the quality of credits that they're using are only high and can be verified. So credit stacking, we're looking at combining blue carbon credits and biodiversity credits in one location, since we will be restoring the seagrass and gaining these benefits at that single location, which makes them co-located. And that means that we can combine them into one transaction where the accounting for blue carbon and biodiversity will be linked, and they can't be unbundled, which would mean that it's open for exploitation. People can sell one and not the other. And we see a lot of exploitation in the carbon market, and a lot of the methods that we've been looking into have really done a lot to evade that kind of exploitation. Looking forward, within 5 to 10 years, aspirationally, we would love to purchase blue carbon credits in the next 1 to 2 years, restore local seagrass meadows beginning in 3 to 4 years, but we understand that restoration may take a little bit longer, and crediting biodiversity beginning in roughly 3 years, since it is such an emerging market. We hope to see results in 5 to 10 years, although again, we understand that's very dependent on when species decide to return to the site. And obviously, we'd love to improve ecosystem health for our vital seagrass meadows as soon as possible. And that's why we want to move forward with the blue carbon credits as soon as we can. With that, I thank you, and I can open it to questions.
We have time for 1 or 2 questions from the audience, if anyone would like to ask. Somebody's got to have something they're curious about here. You guys might get off look easy. Excellent. All right, well, thank you so much, team. Have excellent presentation and great work this year. I know that the program will be very thankful for your results. So thanks so much. Please don't be shy with questions after presentations. You know, put them to the test. They might have made all this up. You don't know.
Can I, the team can come outside?
Oh yeah, yes.
To take a photo, or should I wait until the break?
I would say do it after each presentation. Thanks so much. Next, we have a team that has worked with Critical Ecology Labs on the ecological legacy of slavery on plantations in St. Croix.
Right. Hello, everyone. My name is Miranda Hess, and these are my co-speakers, Ronnie McChesney and Peter Jensen. And we've been working with our client, the Critical Ecology Lab, to research the ecological legacy of slavery and plantations on the island of St. Croix in the US Virgin Islands. So St. Croix was used for the intense agricultural production of primarily sugarcane for more than 400 years. Unjust social systems like slavery and plantations, which were used for this agriculture, can leave behind ecological scars on the land. And social and environmental equity are closely intertwined both in the past as well as today. The primary question that was posed to us by the Critical Ecology Lab was, how have Danish colonial plantations impacted secondary forest regrowth and soil health on St. Croix today? So our research delved more into the historical variables of land use intensity, the viability of archival soil versus fresh soil samples taken from St. Croix, And finally, the way that soil samples vary depending on whether the land was used for agriculture or not. The results of our findings can then be used for future environmental and social equity problems. For historical data collection, we focused on historical and archival documents with three main variables: windmills, enslaved population, and finally, sugar production. These three variables would then show us a picture of how intensely the land has been used. We used two main sources for two of those variables, one of which we looked at head counts of enslaved populations from tax lists, and then we also looked at sugar production on documents from the Royal Danish Library. Unfortunately, some plantations had gaps in data for particular years, and so not all plantations could be compared against each other equally.
And so here's a map showing the plantation boundaries for St. Croix. This will provide some context for the next few slides, and just so you guys get a scope of how many plantations there were on the island. The first factor that we're taking into consideration when estimating how intensive the land was used are windmills, as these were used to produce sugar from sugar cane. And so we chose to use three different sources to discover windmill locations in order to account for changes in plantation boundaries over time and the additions of more windmills. And so we calculated average density of windmills per acre for each of these three sources, and then added those together to create this map that you see here. The darker red indicates a higher average windmill density per acre and therefore more intense use of the land. One thing you might be wondering is why we didn't calculate the average windmill density per acre for each of the individual plantations, and this is because one of the sources we used was a historic map from the 1700s, and it was relatively geographically inaccurate. The locations were off a little bit of the windmills, and so we decided to remove the plantation boundaries when calculating windmill density in order to account for these geographic inaccuracies. The second factor that we took into consideration when estimating land use intensity is average sugar production. We selected 5 estates to gather the sugar production data for. These are taken from a list of estates that our client was gathering soil samples for, and we only selected 5 because historic data collection is very time intensive, and we could only look into a few. Sugar production is likely the most important metric when estimating land use intensity, because it directly correlates to the sugarcane that was grown on the island in the soil. The third and final factor that we took into consideration when estimating land use intensity is enslaved population. The top map here shows the average enslaved population for a subset of estates on the island, but the bottom map shows the average enslaved population per acre for each of those plantations. And we chose to do it per acre because this accounts for the fact that, of course, larger plantations will likely have a larger population. And so we used that bottom map when estimating the final land use intensity, as we can now see here. And so this shows the culmination of those 3 factors that I just mentioned in the final map, with a scale from 1 to 10 of land use intensity, 1 being the lowest intensely used, and 10 being the highest intense use. As you can see on the map, Little Princess saw the most intense use of the land, and Concordia East saw the least. But one thing I do want you guys to note is that we don't intend for this to be a final product for our client. Rather, we want it to be a base from them to build off of as they continue to gather data for more extensive timeframes and for more plantations in the future.
Okay, so just moving on to the second part of our research, we focused on analyzing various components of soil chemistry as they might relate to land use intensity. And ultimately, this data is actually going to be paired with that spatial component that Anahita just discussed, and we'll be able to either confirm or reject some of the predictions that we made with our land use intensity index. So we began by extracting eDNA from soil samples collected from the island of St. Croix Estate Little Princess in 2022 and collected here on campus this year. So eDNA, which is DNA that's left behind by various soil inhabitants, such as bacteria, such as fungi, can tell us a lot about soil microbiome health, which in turn can tell us a lot about ecosystem health. And the samples we analyze also have the potential to tell us about the differences between soil quality in old growth forests and secondary growth forests, where old growth forests are those that have been left undisturbed for an extended period of time, and secondary growth forests are those that have been disturbed more recently, so by plantations during the colonial period. So the first step in our analysis was to extract eDNA from both the archival and the fresh soil samples. And these are some of our findings right here. First, we found that the average eDNA concentration in the archival soil samples was actually higher than the fresh soil samples. And then we also found that the average eDNA concentration in old growth samples was higher than that in secondary growth samples. So what this tells us is that archival soils are actually a viable method for extracting eDNA from soils, and also that sugar cultivation may still be having an impact on the soil health today. And then following DNA quantification, we also used a specific primer to isolate and amplify bacterial eDNA extracted from the samples using a polymerase chain reaction. And then following this, we used gel electrophoresis, which essentially just separates these fragments out based on size in order to ensure that we actually amplified the correct region of the DNA. And then we're currently in the process of sequencing the DNA amplified during the PCR, which is basically just going to tell us a little bit more about the microbial diversity in old growth and secondary growth forests across the island. And then ultimately what this will do is provide us with information on how or if the ecological impacts of sugarcane operations made possible by systems such as slavery are still visible across the island today. Okay, so in summary, our team developed a methodology which synthesized historic data using spatial analysis in order to come up with a land use intensity index using the variables that Anahita mentioned, which were windmill density, average sugarcane production, and then enslaved population data. And then this methodology is replicable and is validated by a comparison between soils collected from Estate Little Princess and Concordia East could be used to predict how the ecological impacts of slavery are still felt across the island today.
Finally, we wanted to mention that in terms of outreach, we have had our research published in an article written by one of our team members, Maria Almeida, for the UCLA Femme Magazine's June edition. And we also have a story map about Estate Little Princess by one of our other members, Chloe Clegg. We'd like to thank these people for their contributions to our project. And we'd like to thank you very much for your time.
Do you have questions for this team on their research or the work they've done? So in the back, I see
It's a curiosity question. How deep did you go in the soil to get the samples?
So we actually didn't personally take the samples ourselves.
I get that, but how deep did they go to get these samples?
Yeah, I'm actually personally not sure. I know initially we were actually planning on sampling leaf litter, so a very kind of topsoil layer. So I can't imagine that we went much deeper than that. But yeah, unfortunately, we didn't actually collect the samples, so we don't have that entire source of information.
I'll actually ask a question. It doesn't look like there was much of the island that was undisturbed, but compared to what you would expect from sort of primary growth forest, what sort of differences do you see in the secondary growth that's visible there now, or how would this compare to other islands that didn't see as intense agriculture? If anyone has an answer to that.
I think that our findings were more about providing context for what the critical ecology lab will determine in the future about the secondary forests. We did use remotely sensed imagery to take a look at the vegetation health on the island, but we have not yet provided any final conclusions for the critical ecology lab.
Thank you, Raja. Again, nice job. Excellent research, and thank you so much for the presentation.
Thank you.
All right, next we have a team that is presenting for Socially Responsible Agriculture Project and Farm Sanctuary. Come on up, team, and start your presentation.
Hello, everyone. Thank you for being here today. Me and Camille will be representing our team in discussing our project. So, in partnership with the Socially Responsible Agriculture Project and Farm Sanctuary, our team aimed to study environmental antibiotic resistance in waters near concentrated animal feeding operations in Michigan. So, samples from our project were collected from swine and dairy CAFOs, or concentrated animal feeding operations, to detect ARG pollution in proximal communities. Farmers in these CAFOs feed their livestock low doses of antibiotics in order to prevent the spread of disease. However, frequent use of these antibiotics can cause microbiota within livestock to develop an immunity to these medicines, and this can be very harmful. This process is done and happening through ARGs, or antibiotic resistant genes that these microbiota develop. So these microbiota in ARGs and such are not contained in their environment. They can actually spread and contaminate surrounding areas. And we're using a swine manure livestock example to show this case. So on these swine manure lagoon spray field systems, CAFOs farmers use manure in their spray fields to help their crops grow. However, runoff and leaching can cause this manure, which can contain ARGs, to spread into the environments through our water systems, or they can contaminate produce. And as we can see, we can ingest those things, and our bodies can develop an antibiotic resistance. With this in mind, we developed some research questions. Number one, how do we compare results from our 3 different methods that we will discuss to understand environmental ARGs near CAFOs? And number two, how can we use microbial source tracking to identify the source of ARGs near CAFOs? So while we worked really hard to answer these questions, we've come towards some kind of a goal where we want to make sure that our work can push towards support of standardizing a method of ARG monitoring. We work with Lynn Henning, who is a part of SRAP, which is one of our nonprofit partners. And she helped us identify 9 different polluted sites for us to collect our samples from. And our polluted sites, you can see with swine and dairy, we have our swine and dairy to indicate where those sites are in Michigan. And we also have 2 different control sites. One is a wastewater treatment plant, and one is an area not affected by agricultural farming practices. At these sites, we collected our samples using a water sampler, which was designed to have a PVC pipe frame filled with rocks. And those rocks weighted our frames so that empty bottles can sink into our sites that we collect our water from. You can see there's a metal ring where our sampling bottles will be fastened. And there's rope tied at the top so that we are capable of lowering our sampler into water from different heights, as you can see in this photo here.
We've been using a combination of 3 different methods to analyze for antibiotic resistance in our samples. So qPCR and metagenomics are the traditional methods used to monitor antibiotic resistance in water. Both of these methods do have their flaws though. They're very expensive and time consuming, and they also require specific lab skills and equipment. Something unique about this project is that we're trying to incorporate the use of IDEX. IDEX is a method that's less commonly used, especially in the more novel way we've been using it in this project, where we're culturing samples both with and without the addition of antibiotics. This culture-based method is really quick to yield results, which is why it's a good use as an indicator. But combined with the qPCR and metagenomic methods, we're able to get a really comprehensive analysis of the environmental resistance. So IDEX is a culture-based method which can determine the total concentration of E. coli as well as total coliform. And also by comparing cultures that were cultured with and without antibiotics, we can determine the percentage of resistant total coliform and E. coli. qPCR is a molecular-based method where we pre-select specific primers of ARGs and MST genes to determine the concentration of those genes within a sample. It's really important that we do MST or microbial source tracking analysis as well, because these are genes that are specific to the gut biome of specific animal species, so they can help to establish that CAFOs are in fact the source of the contamination. And then lastly, metagenomics is a computer-based method where we send extracted DNA to an external lab for sequencing. And then with this sequencing, we can compare to existing online databases to determine the identity and quantity of antibiotic resistance genes.
And also we can taxonomically identify the bacteria in our water samples. So each of these methods though do have their limitations. For example, IDEX can detect viable but non-culturable cells, or it cannot detect viable non-culturable cells. And then qPCR, because it requires pre-selection, it can't detect ARGs that are existent in the sample unless we've pre-selected them. And also it cannot differentiate between viable versus non-viable cells, and that's because all living cells are lysed during DNA extraction, which precedes qPCR. And then for metagenomics, there is a potential for lower sensitivity, and that is because it's assessing the entire genome. And also, if there's variability in online databases, that can be reflected in the results because we are comparing to those existing databases. So now to get into our results, these two graphs shows IDEX results from samples that were not cultured with antibiotics. So as you can see, there's great variability of contamination across sites. However, each site has a similarly contaminated read between fall and spring samples. Here are results from IDEX that show the percent of resistant total coliform on the left and then the percent of resistant E. coli on the right. These show that spring had a greater concentration of antibiotic resistance genes. A reason for this could be because we collected samples near calving season in the spring. These qPCR results also show the similar pattern of increased concentration of ARGs in the spring versus the fall. And then also, as you can see, we have MSTs on the spring qPCR heat map, and those show that PIG2BAC, which is a microbial source tracking gene, was less present in site 10, which is our negative control site. So that is indicating that CAFO are a source. This is a more specific way that we can compare metagenomics and qPCR. As you can see, site 7 has low risk score and also low antibiotic resistance genes. And then this metagenomic map, each color represents a gene that's resistant to a different kind of antibiotic. As you can see, there's a lot of different colors, so there's a lot of different antibiotic resistance genes. And then the purple represents genes resistant to bacitracin. This is significant because bacitracin is a gene that's used as a growth promoter. So it emphasizes that CAFOs use antibiotics not only for disease prevention, but also to promote growth within their animals. And then our last result is a bacterial community analysis. As you can see by the different colors, we have a different bacteria community across impacted sites, which are on the left, and then the two control sites on the right. This indicates that CAFOs, again, are the source of the contamination, or at least definitely changing the bacterial community.
I know we just presented a lot of results, but it is our hope that our results can help contribute to a standardization of methods for assessing environmental resistome in water samples. Currently, there is a lack of policies in CAFO-related ARG pollution, and this doesn't really encourage a lot of monitoring. So we hope that our results can change that. A protocol that combines IDEX as a primary screening tool and using qPCR and metagenomic methods can help provide a more comprehensive analysis of ARG presence. And that is what we hope to accomplish by the end of this quarter. We wanted to acknowledge a lot of people who helped us throughout this entire process. Thank you to the Joan Doran Family Foundation for your generous support, for Judy Bauer, president of Farm Sanctuary, for Lynn Heading, who identified a lot of our sites in Michigan, and for our advisor, Dr. Jennifer J. and Yue Kong from UCLA's Department of Civil and Environmental Engineering. Thank you so much.
Do you have any questions from the audience? Oh, couple.
No.
I'm gonna skip Jenny for a second. Go to the back. Oh, hold on for Jenny, but yes, in the back.
You said one of the control sites was not like agriculturally contaminated. Given like that we see that there's like a runoff and all of this from these sites, how are you sure that that site isn't contaminated from agricultural spots from like maybe not adjacent but even further away?
Yes, that's a good question. So this site was upstream from the CAFO related site, so that's a good indicator that it would be relatively unimpacted. I do suppose it's possible if there were CAFOs a lot farther upstream, there would have been an impact, but based on our results, we were able to tell that this was a lot less contaminated, which shows that choosing an upstream site was a good choice for a control group.
Now I'm gonna, now that they're warmed up, I'll let Jenny ask a question.
I don't have the question. I'm just saying a comment that you guys are amazing. And I wanted everyone to know how difficult these techniques are. The qPCR and the metagenomics are really grad PhD level techniques that you guys mastered within 1 year. So I'm blown away.
Any other questions from the audience? Great group today. All right, warm yourselves up. You should be peppering these students. Don't get them that easily. Thank you so much, team. Excellent work. It's been fun. So moving from the waters of Michigan, we're going to cross halfway across the globe right now, and we have two projects that were brought to us by AC Milan, the storied football club. And before we get into this, I do want to thank AC Milan's client as our client, who I believe are hopefully watching this presentation up very, very late at night. So thank you again for your support. With that, I'll turn it over to this absolutely fantastic team. Thanks so much.
Hi, everyone. We are the AC Milan Life Cycle Assessment Team, and for our research project, we calculated the life cycle carbon footprint of a soccer match with AC Milan.
And so sport has a vital role to play in tackling climate change. It has an unrivaled capacity to be an advocate and lead by example. With around 5 billion fans worldwide and over 250 million players across 200 countries, football is the most popular sport in the world. The sheer volume and reach of football greatly impacts the environment. It is estimated that the football industry produces over 30 million tons of CO2 each year, which is the same size as the total emissions of Denmark. San Siro, AC Milan Stadium, hosts around 2 million spectators each year, with an average attendance of 70,000. In collaboration with this team, we sought to understand the environmental impacts that result from individual football matches in order to provide recommendations for emission reductions.
So we posed the research question, what is the environmental burden associated with the single football match played by AC Milan? And to answer this question, we synthesized existing literature on sustainability in soccer matches. We then created a life cycle assessment model based on San Siro Stadium, and then we utilized this model to help us identify hotspots and develop recommendations for emissions reduction priorities. And so we evaluated a total of 6 different sectors to conduct a life cycle assessment of a single match day. And so these 6 sectors included field operations, electricity to light up the field, transportation of various spectators, merchandise, heating operations, and food and beverage services.
Our team created this baseline model in order to show the sector breakdown of, or the carbon dioxide emissions by sector activity. We found that carbon emissions for a single match day equaled 214 metric tons of carbon dioxide, and that transportation and food and beverage sectors accounted for the majority of this. These 2 key takeaways allowed us to better dive into which sectors to look into and what would best allow us to decrease these emissions for a single match game.
In our project, each sector was further broken down into the relevant activities. For brevity, we're only going to go over the breakdown for the transportation sector, but the methodology was used across all sector activities. For transportation, away and home team travel only accounted for 0.3% of the share of emission total for transportation, and so we only focus on spectator travel. Based on assumptions of travel behavior, we accounted for 30% of spectators traveling by car, 40% by urban rail, 20% by bus, and 10% by train. As shown in the figure, spectators traveling by car account for the largest share of emissions in the transportation sector, with 47%, even though only a third of spectators take this mode of transportation. Based on the knowledge that spectator cars contribute significantly to the footprint of a single match, we conducted a sensitivity analysis to determine how match emissions might change depending on the percentage of fans traveling by car. As shown in this table, in the green, if you were to reduce the number of spectators traveling by car to 10%, this would result in a 4% reduction in total match day emissions. Even if it were only reduced to 20%, This would still result in a 2% reduction, which accounts for 11 tons of carbon dioxide. This is why it's crucial that sport clubs incentivize fans to travel by public transportation and, more importantly, lead by example.
So for this section, I'll be talking about various emission reduction scenarios for food lounges in particular within the food and beverage sector. So this table in general shows the percent change in food and beverage sector emissions when we vary the percentage of fans visiting and when we vary the percentage of agricultural production emissions. So just for context, agricultural production accounts for the environmental impact of one catered meal, and it can vary, increase or decrease depending on what sustainability initiatives are implemented. So we're going to focus on the far left column over here in bold, and it is shown that if we decrease agricultural production emissions by 20%, we can achieve a reduction in food and beverage sector emissions by around 0.79% up to 10.92%. So this is basically just saying if we hone in on this sector in terms of sustainability initiatives, there's a lot of potential to reduce carbon emissions. So more specific recommendations for this section would look like sourcing from various farms or suppliers that utilize more sustainable farming methods such as crop rotation or greener fertilizers.
Since most heating and electric units stay operable for the duration of a match, we found it best to look at changing the electricity emission factor for a single match. We found that decreasing the emission factor by 10% allowed us to see a 1.3 metric ton decrease in overall carbon emissions. This may not seem like a lot compared to transportation. However, we found it significant considering that no usage habits have been changed, just the reduction in the emission factor. And this can be achieved through sourcing cleaner natural gas and electric sources, as well as using technology or investing in technology that is more efficient for the stadium.
Moving to the field maintenance sector, we looked both at the production of hybrid grass turf as well as the halogen lighting system that is used to promote grass growth. And based on our research, we found that LED lights can reduce emissions by 90% compared to the incandescent lights that are used currently. And so looking at our sensitivity analysis, the bottom part of that table would be our scenario where LED lights are used instead, and it would reduce the share of total emissions from field maintenance from 1.94% all the way down to 0.19%. Then for the merchandise sector, we looked at both polyester and cotton products because the majority of merchandise items sold are made from those two materials. Our research found that recycled polyester can reduce between 25 and 75% of emissions based on the technology. And similarly, cotton can reduce 2.2 to 8.6%. And so looking at a few different scenarios, we found that using both recycled materials can change the share in total emissions by almost 2%.
So lastly, we hope that our findings spread awareness about sustainability within the sports industry, and we hope that our results help inform our client to implement more sustainable initiatives in the near future. A special thank you to the IoES department and to Giovanni Rosati and our project advisor, Faraj Sawan. Thank you so much.
Questions from the audience? Yeah.
If they, if FC Milan were to like implement all of the suggestions, what's like a realistic estimate of how much like total carbon dioxide or like percentage wise, how much could they like reduce in their carbon footprint?
We actually didn't estimate our results in that way. We more did it in a breakdown of like relative sector activities and percentage reductions. I think that would be a great thing to figure out. We just did not produce our results in that way.
Yeah.
Was there any consideration to look at water usage like in bathrooms or sectors?
There was definitely consideration to do that. Courtney and Yara over there were the people who worked on our electricity section, and water and waste production were excluded from our results. just because of difficulty in determining those factors outside of these. I think that would be a great area for further study, but the scope of our project was already so massive, we had to keep it to the sectors that we did.
Thank you so much, Steve. That was excellent. All right, we're going to continue with AC Milan, who were generous in allowing us to conduct 2 projects, research them, but we're going to switch this time to looking at how fan involvement with the team can affect sustainability. So with our next team, please come on up.
Hello, everyone, and thank you all for coming. We are the AC Milan Fan Awareness and Sustainability Team. We were in charge of completing fan-focused research, which we obtained through surveying dedicated AC Milan fans. While the last team talked about the direct actions of the club, we want to focus on the heart and soul of the club, the fans. Football and sports in general is a huge global empire with a monumental effect on the environment. AC Milan, being one of the world's largest clubs with 7 European championships, and a fan base of 500 million, came here to UCLA in order to understand more about their fans' current perception of and response to sustainability. Having the privilege of being able to gauge an ideology like sustainability could have a huge impact on how sports organizations move toward a more sustainable future. I am standing here with our wonderful team who put countless hours of hard work and dedication into this project. Our advisor, Emma, who facilitated all of our team meetings, was paramount in creating the best possible working environment for us to thrive in. We also want to give a special thanks to our liaison, Giovanni, who throughout the year was able to maintain clear communication between our parties, allowing us to seamlessly get to our desired finish line. As for the project overview, our team began the year researching everything from European circular economies to strategies to engage sports fans. This research was the foundation in which we were able to write and develop a survey. This newfound perception of an eco-friendly approach to sports allowed us to then propose various ways in which AC Milan can engage their fan base while continuing to lead the way in sustainability. To understand fan perceptions and attitudes, we conducted a survey that attempted to answer these questions. The first one being, what practices or models exist for linking fan cultures and sustainability initiatives? We used our survey data to measure how fans react to current sustainability practices within their team and ways in which that can be increased using marketing strategies. Our second question hints at the fans' current application of environmentally friendly practices within their lives as individuals and as a fan. Our final research question is an overarching attempt to deliver our client with tangible ways in which sustainability can be applied to their organization with emphasis on productivity.
After we developed and finalized our survey using Qualtrics software, we translated it into an Italian language version as well as the English version that we had written. We sent it out through AC Milan to their fan club mailing lists, and after around a month, we got 250 Italian language responses and 50 English language responses. Because of the lower amount of English language responses, the responses were all analyzed together, as our main point of analysis was not which language the fans speak, but rather their dedication to the club and their regular attendance. Having the survey sent out only to the super fans accounts for the dedication. And then our analysis looked at season ticket holders and other frequent match attendees to account for the frequent attendance. Looking at the demographics of our respondents, those that filled out the survey had an average age of around 40 years old. Around 50% are season ticket holders, and the majority had attended multiple games this past season. An important thing that we were able to establish was that the respondents' baseline attitude towards sustainability was positive. We asked them to agree with the following statement. Environmental sustainability, the idea that humans should live in a manner that conserves Earth's natural resources, is important to me. 97% of the respondents indicated agreement with this statement, of which 77% indicated strong agreement. One of our first key findings from the analysis concerns the sustainability partnerships AC Milan currently has. They have a partnership with Aqua to recycle single-use plastic water bottles and a partnership with Puma to use recycled fibers in some AC Milan branded jerseys and merch lines. We discovered that the majority of respondents to the survey were not aware of either partnership, and only a quarter were aware of both. However, once made aware of the partnerships, the vast majority of respondents supported both of these partnerships. This leads us to propose to AC Milan both an increase in marketing around the existing partnerships due to the low awareness, as well as increasing and expanding the partnerships given the high support. We also looked at campaigns elsewhere to see if AC Milan fans would support similar practices here at home. For example, Portland's NBA team has an annual green game where attendees are invited to learn about sustainability practices and innovations. Another observed campaign is the 2022 World Cup in Qatar, where FIFA attempted to reduce the use of single-use plastic water bottles by increasing the number of water fountains and reusable water bottles. When asked if they would support similar initiatives to these, respondents indicated a high amount of support. So we recommend that AC Milan consider campaigns like having green games, increasing water fountains, and reducing the availability of single-use plastics and promoting reusable containers, so that they can both increase fan awareness around sustainability, as well as materially reduce the environmental impacts, as we saw with the previous team's results.
To understand how AC Milan can best use marketing strategies to engage their fan base, we asked fans to rank their opinion on who they would be most likely to support in an environmental sustainability campaign. The options were the team as a whole, individual players, team management, and other fans. We had hypothesized that individual players would be the most likely to be influential in an initiative such as this. However, we found that in fact, the whole team is the most important to consider. This led us to propose the team using the whole team to be the leadership and face of sustainability campaigns to best engage their fans. We also asked fans to rank their opinion on what they feel a football player's role should involve. We had hypothesized, naturally, that the top two choices would be winning a club team trophy and a national team trophy, and found that the club team trophy, in this case for AC Milan, was the most supported thing a football player should do. Between these two options, fans also ranked that they believe that visiting children in a local hospital is also an extremely important thing a football player should do. This highlights the social priorities of fans and led us to propose combining social and sustainability causes to better engage fans who would otherwise be uninterested. This presentation could not reflect the full extent or detail of our full deliverables. And these are our final deliverables, which make up the culmination of our year's work. We propose ways for fans to increase interactions in sustainability on game day, increase fan awareness, and provide marketing strategies for AC Milan to do this. We could only exhibit a few key findings today and have more detail of our proposals and suggestions in our final paper. When football fans are engaged in sustainability at their clubs, they become engaged in sustainability throughout their lives, which can include billions of people, creating a more green world for all of us. Thank you so much for listening and coming today. Any questions?
Questions from the audience? I assume some of you are sports fans, so. I'm not letting them off the stage until you ask them something. What do you have? Yes.
So you mentioned that you said you would like to combine social and then sustainable. So you engage with fans. What are some examples?
So examples of things that the team already does that's quite social is they do go to children's hospitals, and it's kind of about putting on events that can create sustainable initiatives in a form that is social. So in Milan, there's really terrible air quality, which affects everybody that lives there.
So by making it a social issue rather than like an environmental politicized issue in Italy, it can be more accessible to most people and really influence more people.
Thank you again, team. Actually, wonderful. And thank you again to AC Milan for bringing these two projects to us. Hope it's not too late at night for you and you actually get some rest at this point. All right, thanks so much, team. We are going to maintain a global focus moving to our next project here, but with a sort of a home base or hub right here in Los Angeles. And we have a team coming to work with us from Sky Chefs to tell us about the sustainability and repurposing of airline food in the airline industry. And for our client again, Sky Chefs, thank you for bringing this to us.
Hi, everyone. Our team has been working with LSU Sky Chef all year on managing and repurposing airline catering foods. My name is Joyce. This is Erin, Kevin, and the rest of our team, and our advisor, Professor Deepak Rajagopal. Our client Sky Chef is one of the world's largest airline catering companies. They provide millions of meals for thousands of flights and millions of passengers. Okay, so I want the audience to picture this. You're on a plane and you're served a meal, but you don't finish it. Eventually, a flight attendant takes away your partially eaten meal. Have you ever thought about where that food goes? If your answer is no, well, you're not alone. This issue is very commonly overlooked. What happens is that every day, the packaging and the unfinished meals accumulate into 5.4 tons of waste for Sky Chefs alone. That's 5 times the amount of daily waste produced in all of UCLA dining halls combined. Food waste is such a pressing environmental issue because not only are all the resources being used to produce the food being wasted, it also creates millions of tons of greenhouse gases. So what can we do about that waste? Well, currently all of that waste is just being sent to landfills, but we want to reduce the amount of waste being produced, as well as repurpose the waste in a way that's both beneficial to our clients and the environment.
So our project was comprised of a couple key components throughout the year. Our first job was to make multiple facility visits in which we sampled international and domestic flights coming inbound from different Sky Chefs facilities. And we built key estimates of food waste by going through each individual food cart, weighing the food. I'll go more into that later, but it was a pretty rigorous process. Then we conducted a life cycle assessment based on these estimates we drew from food waste to determine the carbon footprint of the facility from emissions directly associated with this food waste. Then we explored feasible diversion pathways for this waste produced based on logistical, financial, and regulatory considerations deemed material to the facility, both from our initial research and just that we discovered ongoing throughout the process. And then finally, after conducting industry outreach to dozens of waste repurposing players in the region, additional analyses and lots of research, both for pathways that we explored, we proposed a pathway that we deemed to be most feasible and most impactful to our client, both environmentally and financially. Now, the manual waste sorting process was interesting. I'm just gonna go over this a little bit. You know those carts that you see in the planes? We went through each one of those carts coming off of those flights. We pulled each tray, pulled each plate, and we sorted the food ingredient by ingredient into separate bins, vegetable, fruit, protein, carb, to get a better idea of what was being produced, both categorically and more broadly speaking. We used manual entry into sheets. We used an artificial intelligence classification system, and we used the sorting methodology to kind of build estimates that we could then use to represent their daily food waste coming from all their 150 plus flights. So if you look at this was kind of the data that we collected from our sampling. So for all food wasted, this goes from anything from a couple of cherry tomatoes left on a plate to an entire filet mignon meal. 75% of food wasted was just entirely untouched. And from that, we saw that about 30% was grains, 30% was vegetable, 20% was protein, 14% was fruit. These numbers will make more sense in the coming slides, but they helped us determine what pathways to pursue in this food waste repurposing.
So a life cycle assessment, or LCA, it looks at the environmental effects of a product from its raw material extraction to its disposal and everything in between. So with the data we collected, we conducted an LCA literature review of the 4 food categories: proteins, fruits, vegetables, and grains. and this allows us to estimate the carbon footprint of the total food waste. We looked at about 100 LCA studies, and each study gave us a value that represented the amount of carbon dioxide emitted per kilogram of food produced. We averaged these numbers and disregarding any potential outliers like the ones highlighted in red. And then we calculated a weighted average using the category percentages from the previous slide. And we found that for every kilogram of food wasted, it resulted in a carbon footprint of about 3.75 kilograms of CO2. Proteins, especially beef, contributed the most, and cutting beef consumption in half would reduce the overall carbon footprint by about 26%.
And then, as previously mentioned earlier, Bill decided that currently the best option for Sky Chefs to take is animal feed manufacturing. Currently, Sky Chefs process is on the top left, yellow one. They use an autoclave to directly process their food waste, which is a steam sterilizer, which eliminates any risk of bacteria and pathogen contamination. However, we looked into several different options for how they can modify their operation to make the food waste more valuable or easier to facilitate towards animal feed. Our first option is that Reconserve, a company we've reached out to who recycles bakery and grain waste into animal feed. They take the waste directly. Our second option is we use the autoclave and the addition of a dehydrator. A dehydrator is another type of food processing equipment which reduces the moisture content and also shreds the food waste, reducing its volume. And this is a particularly interesting option because the main constraint in the animal feed pathway is that you want a food type with low moisture content. And our third option is to directly replace the autoclave with a dehydrator. However, under current regulations, this is not allowed. However, we think it's something that should be further researched and looked into as a result of our project. And finally, our last option is that Sky Chefs ships their food waste to a dehydrator service, and then from there, we will be able to transport it back to Reconserve. And then, so a few of the recommendations that we decided upon were, first off, for both animal feed and any alternative pathways that we ended up crossing out, such as pet food or composting, a big key point that many industry professionals were interested in is that they want sorted food waste. So we think that Sky Chefs should implement strong protocols to sort their food waste, which would facilitate easier disposal of their waste. And we also believe that Sky Chefs should try a pilot of the Reconserve recycling waste, either in the form of just grains or bakery waste, or both grains and proteins, as these are the two food type categories with the largest carbon footprints, and will reduce and will have the highest effect on reducing the carbon emissions. And then as for our discussion, some of the main takeaways that we found from our project is that, one, using animal feed, it's very easy to replicate across any airline caterers that have access to a grain recycler, because all you need is essentially to sort the waste or the moisture content, and then you can establish a partnership with the company and then send it to them. Also, there's potential to generate a profit from this type of waste disposal, as some animal feed companies are willing to pay a set amount of money to take the animal feed waste, so specifically the carbohydrates and protein waste. And finally, we also believe that our project can be a trailblazer for further changes across the aviation industry, as while we did conclude that at the moment animal feed is the most sustainable option and viable, there's still a lot of research to be done in other sectors such as pet food, and also in terms of trying to improve other forms of processing technology such as the dehydrator to easier facilitate this type of disposal. Thank you. Any questions?
Yeah, a couple of questions. Great. Let's start with Kelly. Yeah.
I was pretty shocked to see that such high percentage of full meals that are wasted, and I was just wondering if you had any thoughts about maybe they just shouldn't make that many in the first place.
So while we tried considering that initially, there are a few limitations because ultimately it is up to the airlines, what the airline catering companies produce. We were hoping that further along, because Sky Chefs plans on doing additional projects with both the Luskin School and probably the IoES as well. We were hoping that at some point we could involve other stakeholders such as airlines, community members, and possibly regulatory bodies, both municipal and federal, to kind of work on reducing this food waste. But at the moment, the best bet is just diverting it because they ultimately have the ultimate say, the airlines.
So another question over on the other side here, I think. Oh, there's one in the middle, yes.
I was wondering if in your analysis of the recycled feed manufacturing process, if consideration for nutrients being fed back into the animals is something taken into consideration, like when you dehydrate and process that food, are you losing nutrients for the animals that you're refeeding back?
Yeah, that's a great question, actually. So we brought that concern into Reconserve because we were kind of unsure about that. We thought it would be a different step in the sorting process, kind of nutrient monitoring. But they said that they didn't seem to indicate that dehydration kind of removed any of the nutrients that they were most concerned about. And they also said that this food waste would be part of a 100-ton daily mix. So whatever nutrient loss could come from this food waste particularly would be insubstantial, but it would still be a great addition to their daily mix. Thank you.
Yeah, nice job, team. Thank you so much. Okay, we're going to take a little bit of a transition here and move to looking at ecosystem biodiversity and some historical ecology questions. And our next project coming up here is with the Nature Conservancy, looking at plant reduction projects for the California Islands.
All right, welcome everyone. We are here to present our project on prioritizing plant reintroduction candidates for the California Islands under future climate projections. The archipelago of islands off the coast of Southern California is a biodiversity hotspot, home to hundreds of endemic vascular plant species. The cluster of 18 islands and proximal regions between San Francisco and Baja California is part of the California Floristic Province, for which our research has substantial implications. Humans have facilitated significant environmental degradation here, and the damage is expected to worsen as climate change becomes more invariable and extreme. Introduced vertebrates like sheep and pigs overgrazed and severely disfigured natural systems, landscape features, and ecological services. As a result, 3/4 of the California Islands' original biodiversity has been lost, and the quality of the land has been severely degraded as a result. To rehabilitate the rich endemism so historically prominent on these island habitats, the Nature Conservancy advocates for a core shift in the reintroduction strategy from extirpated endangered species to extirpated regionally common ones. This has important implications for increasing the overall biodiversity of the California Islands and rehabilitating these diminished ecosystems. Our research question: how can we strategically prioritize the reintroduction of extirpated plant species on the California Islands? Attempts to explore this problem. To answer our research question, we conducted a literature review on reintroduction methods and species extirpation, which is also known as a local extinction on the island territories.
engaged in interviews with island managers and ecologists, evaluated the characteristics of vascular plant species to inform prioritization criteria, modeled species fitness through a combination of mapping and habitat suitability modeling, and tested a decision framework island managers can use to select a viable candidate for reintroduction.
So after producing our literature review, our team uncovered several knowledge gaps relating to our study topic. And in order to fill in these gaps, conducted 15 interviews with island managers and industry professionals from 9 different agencies across the California Floristic Province. These interviews provided valuable insight into the ways in which land managers are approaching plant reintroductions. And some key findings were that it is extremely important to consider multiple ecological factors when evaluating a species for reintroduction, and also that there is a great need for increased resources on how to pull off these reintroductions. At this point, after producing our literature review and conducting our interviews, our team now had a well-rounded understanding of extirpated plant reintroduction efforts across the California islands, and were able to switch focus to the production of our LEAF model, which is a species reintroduction prioritization scheme that will be able to be used by land managers across the California islands to aid in determining extirpated species for reintroduction. After a lot of back and forth and many hours, our team eventually settled on a conceptual question-based model with 2 phases. The first of these phases, the screening section, aims to determine if a species is a good candidate for reintroduction at the most general level through a series of hard stop checkpoint questions that serve to quickly weed out species that do not meet basic requirements, such as being native to the island or having up-to-date data confirming their absence. Our finalized version of this product also includes a sort of user guide with explanations for each question so that users can easily and accurately understand what they're asking. From here, if a species passes the screening phase, it then goes on to the prioritization phase. This section allows land managers to essentially rank species post-screening through a categorized question-based point system with totals that can be compared between species, and is important in that it allows for flexibility in determining which reintroduction factors are most important to a given land manager. This section has 4 categories, the first of which is longevity, and this includes questions relating to a species' odds of surviving and thriving in its reintroduced habitat for an extended period of time. The second category, ecosystem services, looks at the benefits a species brings both to other organisms and its ecosystem as a whole. The third section, added benefits, takes into account the cultural and social values of a species. And the last section, feasibility, accounts for the costs and challenges of reintroducing a species.
To test our model, we used Santa Cruz Island as a case study. We created a list of presumed extirpated species from data given to us by our client, which ended up including a total of 26 different species. In order to test this model, we used research from sources such as online databases and reviewing published scientific literature for specific information about the species, their genus, or their family. In addition, we also used some mapping and habitat suitability modeling to help answer some of the questions.
Habitat suitability modeling, which we conducted in the MaxSense software, supports our LEAF prioritization model. We modeled species distribution under present and future climate conditions in order to understand species suitability and distribution. The study areas that we made these models for were California, the Baja California Peninsula, and the islands of the Californias. Here are 2 habitat suitability models that depict potentially suitable habitats for the round-leafed filary in present and future climate conditions. The map on the right shows a significantly reduced potential habitat for the species in 60 years' time. This was performed for the remainder of our target species project.
The first step in testing our model was the screening phase, which the results can be shown here. 7 of the species did not pass the screening phase, 3 of which were found to be non-native species, 2 of which were found to have recent observations since 1960. The last question of the screening phase had 2 species that were found to have no reliable source populations, which means a population in which the plants or seeds could be sourced for reintroduction. To assist with this question, we created visualizations such as this one here, which is for one of our species, the island rockcress. The next step was testing our prioritization phase, in which there was no species that was clearly superior in points for any of the categories. This is a visualization here of the results of that in terms of percentage of points possible for each of the leaf categories. And here you can see that the highest score that we got was 80% and the lowest score was 0%. This is another visualization of our results by species and the points per different category, with each of the categories being represented by a different color. In terms of overall points gathered, the number one species was the southern honeysuckle, which is shown here. The process of creating this model was a challenging one because we found that there was nothing in published literature about this topic and specifically about common species. What we have created is a new framework to assist in the decision-making process for island managers, which is an important first step in furthering restoration of island ecosystems by reintroducing extirpated common species. We hope that this model can be used to assist in the implementation of restoration efforts on the islands of the Californians and provide an example for similar situations around the world. Thank you for listening to our presentation today. We're happy to answer any questions.
Excellent. Questions now to see one in the middle here. Yeah.
How did you apply your habitat suitability model 60 years in the future?
Evan, you want to take it?
Basically, we used WorldClim, which kind of compiles a bunch of different future climate models through different climate projections like RCP 4.5, RCP 8.5, which is like the intermediate and high emission scenarios, and then like using future climate temperature, future climate maximum temperature, future climate precipitation, and kind of like looking at how that is the present day and then projecting that into the future on like a kind of climatic scale.
Other questions? I'll ask one, which is, so you worked with Nature Conservancy and used Santa Cruz as a test island, as a sense. Do you know what plans would be to sort of coordinate this among the other islands, for potential actually starting looking at action plan for reintroductions?
So the plan essentially is for the Nature Conservancy to use this model for their testing of reintroducing, because none of the islands at the moment are really focusing on the reintroduction of extirpated species. So they're basically first going to test it out and try some actual reintroductions on Santa Cruz Island. And once they've done that, this whole model will be shared with all of the islands so that they can use it either directly or like as a framework to help with their decisions for reintroductions on their own islands. Since every island is basically managed by a different manager, they all have different ideals of like what they want. So yeah.
Thanks so much. Again, congratulations. And looking at management of biodiversity and ecology at a much closer scale, our next team is going to be from working with the city of Santa Monica and looking at potential future development of the current airport site. Come on up.
Hello, everyone. Before we get started, I just want to say on behalf of our entire team that we're feeling very inspired and grateful to be a part of this Practicum cohort class, and very excited to present to you our presentation, Informing the Future of Santa Monica Airport, Scientific Insights and Historical Perspectives. This is our team, advised by Dr. Longcore. Presenting with me today are Yeji, Insu, and Jake. My name is Charlene. And so the Santa Monica Airport, also known as SMO, was first an informal landing strip in World War I, but became an official airport in 1927. In World War II, it became the region's center for economic growth and community pride. But with the introduction of jet planes in the 1950s, it soon became a source of unwanted noise and pollution. After years of litigation and community engagement, the airport will be returned to local control in 2029. This 200-acre plot of land is one of the largest underutilized plots in all of the LA region. As this quote says, it has the potential to be the most transformative urban planning event of the past century for the city of Santa Monica. This site is only 4 miles away from UCLA, and its current plan is to become a great park full of opportunities for education, recreation, and restoration. Therefore, it's really exciting as it expands the accessible green space that we have, and it's very bikeable, busable, and just a 15-minute drive. Knowing the past is essential for creating a sustainable future. So throughout this region, pavement has buried historical streams, bluffs, and native plants. Understanding this history is essential for reimagining a future that's more sustainable and beneficial for the natural ecosystem around us.
Okay, so our project has two primary objectives. So the first one is to perform comprehensive research into the physical, ecological, and human characteristics of the SMO site before it was an airport, and to effectively communicate the landscape history of the SMO to inform site planners. We also went through online databases for any information related to the airport or related terms, and consulted with local institutions to access records of the airport and also surrounding area pre-1930. After compiling a data of archive maps, photos, newspapers, and more, we created an ArcGIS story map to tell the ecological history of SMO. And also, if we were to walk from a northern corner of the airport to the present day runway, we would have to cross the stream bed and climb an elevated bluff to reach the top. It may have looked something like this. The bluff is much flatter now. And also look at the historical property maps like this one from 1888 of Rancho La Bayona. We can confirm the existence of a bluff at SMO. It is overlaid on a modern map to show where the airport was constructed. This historical topography forms the foundation from which we can understand water flows, soil conditions, and life forms that once called the region home. And this is an image of Santa Monica Airport that was taken in the 1950s. As you can see, the bluff we saw in the Rancho La Bayona has been purposely flattened to build a runway, removing most of the local ecosystem of the area. It makes sense that this site was chosen for an airport due to its high vantage point. And on the southern edge of the airport, you can still see some small canyons and alluvial action due to natural drainage that had not yet been lost.
Understanding historical water flows through the region is important for providing environmental context and for illustrating the change over time for the SMO site. We created a digitized waterways map using USGS topographic historical maps. The SMO site is the shape at the center of the image. It's at the western edge of the Bayona Creek watershed. Ephemeral streams used to flow throughout the region very dynamically because they were free to meander. One stream actually crossed the northern corner of the site, and water from the top of the bluff would have flown southwest and collected in a vernal pool that's at the site of today's Penmar Golf Course. Here is a topographic map from 1925. You can see the dotted blue line is that stream that I pointed out next to the aviation field. It was ephemeral, which means it would have filled during cool, wet winters and remained dry for most of the year. We confirmed the existence of this stream using aerial photography. This winding path of erosion and vegetation at the top right of the image would have been the stream's path. By 1934, you can see that the stream is no longer labeled on the map due to expansion of the airport and surrounding neighborhoods. And by 1950, all regional water flows had either been channelized or covered up. Here is an example of how we illustrate these changes over time in our story map. It's a slider with the 1934 and 1950 map. We also looked at soil conditions, as they are important indicators of habitat types and the ability to reintroduce new flora into the future park at the airport. As you can see, it's impossible to see anything through the asphalt that currently covers the runway, so we had to look to historical soils maps and recent boring tests to get a glimpse underneath the airport. This 1916 soils map indicates that most of the soil underneath the SMO site consists of sandy loam, and recent boring tests show that despite profound changes in the airport's appearance and ecology, this soil type largely remains the same. Luckily, the soils map had previously been digitized, and we can see it at a higher resolution, but sandy loam soil has a high water retention capacity and drainage capacity, as well as clay content, which contributed to its ability to support the local coastal sagebrush flora that once inhabited the region.
So based on our research and data from the LA Landscape History Project, we can infer that the Santa Monica Airport area was most likely a coastal sage scrub surrounded by a wet meadow and a large vernal pool. So coastal sage scrubs are a rare biodiverse habitat with only 10% of the original coastal sage scrubs in Southern California remaining. We cross-referenced records from the Consortium of California Herbarium along with other herbarium records and data on coastal sage and vernal pool flora to compile a list of flora that could have potentially been present on the site. So the flora that we see today on the Santa Monica Airport were mostly seeded and were specifically selected not to attract birds. So out of our list, we selected 5 flora to potentially reintroduce to the site, and they were specifically selected for their drought resistance and their ability to attract wildlife such as birds, insects, and small mammals. And so one of our recommendations is chia. Chia is native to the area. It is a staple food source and used as medicine for the tribes of Southern California. And they are also an important source of nectar for pollinators, and they attract small birds, mammals, and insects that feed off its seeds. So some key takeaways and recommendations. We recommend that the airport reintroduces the native plants back into the area and focuses on reintroducing or attracting the birds and pollinators that once lived there. And we know that it is impossible to restore the ecosystem and the landscape to what it originally was, but we believe that these plants will sort of revitalize the ecosystem and ecosystem services, sorry. And another recommendation is to have a learning center that shares the site's historical and ecological history. And we hope that this informs the public on the importance of why we should conserve this site for future generations. Thank you for listening.
Questions from the audience about the airport site or the potential ecology for it? So for those of you who don't know, this project, the Santa Monica Airport site is sort of an unprecedented opportunity where after, as they said, decades of litigation and ongoing process, this is going to be converted from an airport into something new, and the opportunity for park space and ecological restoration and a host of other activities is really astounding. So this work really can have a huge impact on what they're doing. Yeah.
How do you like the name owner of this land? Under control of like the city, was that to offer, there will be like options?
It's getting returned to control of the city. So once it's converted from an airport, it no longer has to follow like all the regulations that an airport has. It can be designated as something else. So, I mean, the decision to return it to something that isn't an airport was made by the city council. So there's like ongoing community engagement with Sasaki got selected as the landscape architecture firm to design the park. But so now they're engaging with the community to like see what they want and having all these meetings to make sure that they're informed.
Are there any sort of remediation efforts that they have to do to like return the or airport to a park because of pollution or that kind of thing?
The soil boring tests that I mentioned on one slide were they were doing testing for pollution, and they found in like one section of the airport there is some lead pollution. But for the most part, like it's just completely covered in like paved surfaces and asphalt. So I don't think they necessarily know everything that's underneath it. We went on a site visit and they mentioned that like there are some cracks in the runway and they like dropped holes down it and they're like deeper than 6 feet. So it's gonna be like Christmas for them. Like,
Potentially contaminated. There's another question over here, yes.
So topography and historical waterways, what is like the plan or like what could potentially happen as far as restoring those?
Those are important for understanding the context of the site. So our role is really just like developing as full of an understanding of the airport's history before it became an airport. So while like they're not going to be able to like rebuild the bluff to what it looked like exactly before the runway was there, or put those streams back where they were, having an informed idea of like where the water might have been moving can sort of help them make this a dynamic landscape again, instead of just some static place where planes land.
Well, thank you so much, Steve. And as a follow to that question, for those of you who know, the center of campus actually is recently as the 1920s is a massive arroyo that runs right down the middle of campus. And I'm still waiting for UCLA to come to the Practicum with a project to daylight that arroyo and restore it fully. So basically in the middle of campus, just a river. They have not taken me up on that offer yet. With that, thank you so much to our teams. Excellent work. We're going to take an intermission and break now for about 20 minutes or so. I would ask the people to be back in the auditorium at about 5 minutes before 3. In the meantime, we have refreshments, drinks outside, take some fresh air, and we'll see you all back here shortly. Thank you so much.